Okay, so we're going to go through the rest of 2.7 today. We talked about replication already, DNA replication. We talked about transcription. Now we're talking about translation. And this is translating the information in RNA into a protein. So just to review, this is kind of like the whole process, right? So here we have our DNA inside the nucleus. Through the process of transcription, we translate or we transcribe the information in the DNA into mRNA. Put DNA up here. That mRNA goes through processing, and after all the introns have been removed and we have only exons left, it exits through the nuclear pore. And it goes out into the cytoplasm. So this whole thing, this process, this is transcription, right? Today we're going to talk about translation, and that's where the mRNA molecule is read by a ribosome. That ribosome then takes the information in that mRNA and uses it to string amino acids together into a polypeptide chain. Now remember, you put polypeptides together and you get a protein. So this is really the process that we're talking about today, and this is translation. If you can draw something like this for yourself, it's a good review. In fact, at the end of this lecture, this is the end lecture for 2.7. It asks you to create a one pager that goes through this entire process. So this diagram right here is a nice overview of that process. So let's talk about the specific understandings. Translation is the synthesis of polypeptides on ribosomes. Now remember, polypeptides are chains of amino acids. You put those together to create proteins. So what we're really talking about here is protein synthesis. Translation is the process of building proteins from information stored in RNA. Now, we're going to go through this entire process in detail, but just as a quick overview, ribosomes are kind of like kindergartners. They can only read 3-letter words. 3 letters at a time, that's it. Cat, dog, boy, bag, stuff like that. Each 3 letters on the mRNA strand is called a codon. So here we have 1, 2, 3, 4, 5, 6 codons. Each codon is a match for the delivery of one specific amino acid. Now remember, the purpose of this is to create a single protein, and a protein is a chain of amino acids. So each codon corresponds to an amino acid. And that really encompasses understanding 6 and understanding 7. The amino acid sequence of polypeptides is determined by mRNA according to the genetic code. Codons of 3 bases on mRNA correspond to 1 amino acid in a polypeptide. Our understandings are over here, just in case. And this is kind of just another example of that diagram that I just had. Here is your mRNA strand, and we have our base pairs. Well, we just have, it's not really a pair, is it? It's just a base. G, C, U. G, C, U. Those 3 letters comprise a single codon. That codon codes for an amino acid, in this case, alanine. Now we're going to go through and practice using an amino acid chart to tell us what codons belong to what amino acid. And we're going to work it backwards too. In fact, that's a skill you have to know. Use a table of genetic code to deduce which codons correspond to which amino acids. Now, something that's important to note, this table, this codon table, is pretty much the same for basically every organism on Earth. And that's because we all speak the same genetic language. We all speak A's, T's, G's, C's, and U's. It doesn't matter if you're eukaryotic or prokaryotic. It doesn't even matter if you're a virus. They're not even alive, and they still speak the same genetic language as us. In fact, that's all they do. So this becomes extremely important when you're talking about genetically modified organisms. This is why we can take genes from one organism and put them in another, and they still work because we all speak the same language. So the prompt is asking to find the AUG codon. So when we're using a square table like this, they're set up so that the first base is located on the left-hand side, the second base is located across the top, and the third base is located along the right. So if we're looking for the AUG codon, I find A, my first base. U is my second base, so I come over and down. AU, and now I look for my third, AUG. I would love to tell your Spanish grade that we all speak the same language, but that's a human thing. That's like a human communication thing. And I don't know, human languages, I only speak one human language, and that's genetic code. That's it. The rest of that language stuff, it's all geopolitical borders. I don't really care so much about that. That's why I'm a science person. Oh, I forgot I'm recording. I was responding to a question or a prompt in the chat. So yeah. Okay, gonna continue. So here we have A, here we have U, and here we have G. Now you'll notice AUG codes for methionine. Now you'll also notice it says start codon right here. This is important. Methionine is the first amino acid when you're generating a protein. It's always the start codon. The start codon tells the ribosome where to start translating. Not all of the DNA in the strand of DNA codes for a gene. Additionally, when you have a strand of mRNA, the coding portion of the mRNA strand always starts with methionine. So, whenever you are translating a protein, you can bet that the first amino acid that it's going to call for is methionine. Additionally, if you're looking at a strand of mRNA, if you spot AUG, that's a good indication that that's where you're going to start. So when a codon reads CCA, C, C, CCA, proline is going to be delivered to the ribosome. And this process continues until you hit a stop codon. Now, there's a couple of different stop codons. There's UAA, UAG, UGA, and I guess that's it. I thought there was one more, but maybe not. That tells the ribosomes, okay, this is where you stop. So now we're going to practice using the amino acid table. I'm going to leave this up on the screen, and I would like you as you individuals, to type in the chat window the different amino acids that go along with these codons. And you don't have to type the whole word. You can just type the abbreviation, right? So go ahead in the chat window. I want you to type all 6, or is it 6? It's 5. All 5 amino acids in order using the symbols. Or if you want to be brave and say it out loud, you can. If not, just put it in the chat window. And I'll give you a few minutes to do that. All right, James says the first one is TYR. Actually, let me put it on the other side. Thanks, James. All right, let's see. Let's see if Amy confers. TYR, LYS, LEU, leucine. So that's what? Tyrosine, lysine, leucine, aspartic acid, and stop. Nice job, guys. I think you have the hang of it. Okay, let's see if you can do the same thing but backwards this time. And I bet you're going to come up with different answers. All right. I'm looking in the chat and I see a bunch of different answers. So for serine, I see lots of people put UCU, but I also see that somebody else put AGC. So let's see. Where's leucine? Where's AGC? We have AGC. AGC is serine, right? All right. 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 All right But we see, let's see, UCU. UCU. So we have a whole block of serine over here, right? Now that's important. I'm glad you guys came up with different answers. The point of this exercise is to show you that there are lots of different ways to spell the same amino acid when you're talking about codons. So while there's 20 amino acids and there's 64 codons, that tells you that not every codon has its own individual amino acids. You have replicates. Now, this becomes important when you're talking about mutations. So, say I have an mRNA strand and I have a codon that's UCG. And for some reason, a mistake is made, and instead of UCG, I get a UCA. Now, the resulting protein is going to look exactly the same because they both code for the same amino acid. So that protein is not going to be misshapen. It'll be able to function just fine because there was no actual change to the protein itself. That allows for a lot of leeway in how you build and how you code the information for specific proteins. You can kind of think of it as like, you know how in the English language we have there, and there, right? Now they all sound the same, but they're spelled different. Or two, and two. You can code for the same amino acid different ways. Oh, I see a question. Why does arginine have two different abbreviations? Let's see, arginine. Let's see. I see AGN. Oh, I see AGN and AGR, or ARG. That's a good question. Arginine. I've only ever seen it this way. I'm not sure what's up with that. Maybe that's to tell you they're doing it differently to give you an indication as to whether the codon starts with a G or an A. Or I'm sorry, a C or an A. That's probably it. I mean, in practice, when I worked in the amino acid geochronology lab at USF, we always used ARG. That's because we were working on the back end, though. We were looking at amino acid concentrations in fossils to date fossils to interpret sea level rise during the Pleistocene. But I think that's because we were only looking at the actual amino acid. We weren't looking at the mRNA codons for it. We were looking at the physical structure. So I guess it doesn't matter which codon you code for. But yeah, my guess is they're using two different abbreviations to give you an indication as to whether it's a codon that starts with an A or a codon that starts with a C. You are not going to have to be responsible for that. It's not going to make a big difference. In fact, you will always be given a codon chart. You will never be asked to name a codon that codes for serine without a table, or you will never be asked to do the vice versa. So you will always be given a codon chart if you need it. Okay, let's keep going. Understanding number 8. Translation depends on complementary base pairing between codons on mRNA and anticodons on tRNA. So just like complementary base pairing works to allow DNA synthesis to happen, right? Complementary base pairing is really the root of the reason why nucleic acids work as information storage at all, because you always have a predictable template by which information is going to be carried. If you have a T, it's always going to go with an A. If you're talking about RNA, it's going to be a U. G always goes with C. Those rules allow genetic language to work. And you'll see what I mean in just a minute. So the ribosome is the thing that strings the amino acids together, right? But they have to get there somehow. The last form of RNA that we're going to talk about is transfer RNA, or tRNA. Transfer RNA is a molecule that fits to the amino acid and it brings the amino acid to the ribosome. So here's our tRNA, and it's going to take that amino acid to a ribosome. Now, each tRNA molecule is specific to a particular amino acid, and it's that specificity that allows the system to work. That specificity relies on complementary base pairing. So that tRNA molecule has something on it called an anticodon that matches the amino acid. So here we have a strand of mRNA. And here's AUG. That's our start codon. It also codes for the amino acid methionine. So it's tRNA's job to go find a molecule or an amino acid of methionine and bring it to the ribosome. Well, how does the tRNA molecule know which amino acid to grab? It knows because it has the anticodon to this specific codon. It has the match, right? A's always goes with U, U always goes with A, G always goes with C. This is how the tRNA molecule knows which amino acid to go get. If I were you, I would draw this picture in your notes. I have seen exam questions that ask you to draw a visual representation of a tRNA molecule. And what they're doing is they're looking for both parts. They're looking for the anticodon, and they're looking to show the attachment site where the amino acid attaches to. So if you are asked a question on an exam that asks you to draw an anticodon, I would draw and label this whole thing. How do you feel about tRNA molecules? Give me a thumbs up if you're good, or a thumbs down if you're not quite sure yet. I see a couple of thumbs up. Okay, let's see, 3. Good. Okay. Here we can see this process in 3 separate stages. So mRNA is red, so this green thing, this is the ribosome. We have our mRNA, and it's being read from left to right. Actually, I think, yeah. This is 3' to 5', so this is 5' to 3'. Okay. Here's our start codon. We know our start codon codes for methionine. This tRNA molecule has already dropped the methionine off, and now it's leaving. The ribosome is currently reading this codon, UGG. That is the codon for tryptophan. So, a tRNA molecule has brought tryptophan and used a condensation reaction to bond that tryptophan to the methionine. Now think back when we talked about our other topic 2 categories. We talked about hydrolysis, which is breaking molecules, and we talked about condensation reactions, which build bigger molecules and water falls out. It's that same reaction that's happening right here. The next codon in the mRNA is UUC. That corresponds to the phenylalanine. Phenylalanine. I'm having trouble with amino acids today. Phenylalanine. There we go. So a tRNA molecule that codes for phenylalanine is going to deliver that phenylalanine and use a condensation reaction to bond it to the tryptophan. And the process continues until the ribosome hits a stop codon. So let's do this ourselves. Now, I'm going to have to rely on you guys to help me because I don't have an amino acid chart or a codon chart in front of me. So you're going to have to use the slides or Google one, because they're all the same, and help me out here. And you can type it in the chat or you can unmute yourself and say it. So here we have a strand of mRNA. And we have our ribosome, and it's reading the first codon. I can do the first one. This is our start codon, AUG. So I know that that codes for methionine. So I'm going to click methionine. Here comes our tRNA. It's going to drop off that methionine. Next, the ribosome is going to read CCA. So now you guys are going to have to tell me what amino acid corresponds to CCA. You can either type it in the chat or you can just tell me. Amy and Hannah both say proline. So let's see. Correct. CCA is the codon for the amino acid proline. tRNA is going to deliver proline. Here it is. And it's going to use a condensation reaction to bond these two together. And then that tRNA is going to leave. Our ribosome is going to progress. And it looks like it went a little too far. We need to code for UAU. Somebody tell me what UAU stands for or what UAU codes for. Amy says tyrosine. Somebody want to back her up? UAU? Yep, Hannah backs her up. Tyrosine. So let's try it. Now I gotta find it. There it is. Good deal. Now, GGC. Amy says glycine. Let's see. Yep. Glycine. Okay, now what happens? We have three choices. DNA helix splits. mRNA reads the DNA. I said DNRA. Dumb. mRNA reads the DNA. Or the tRNA exits. All right, so if I was using process of elimination, we're not talking about DNA at all, right? We're talking about RNA, so it's definitely not this one. mRNA reads the DNA. No, that's transcription, right? There is no DNA. We're like out in the cytoplasm, hanging out with the ribosome. DNA does not exist in this dojo. DNA stays in the nucleus. So that must mean it's this one, tRNA. Surprise. Okay, somebody tell me UAA. My ribosome's getting ahead of itself. We should be doing UAA. Hannah says there are 2. Yeah, there's 2 codons. We're looking for just UAA right now. It's a stop codon for UAA. Okay, so let's see. So I hit stop. Oh, Hannah says for glycine. There's 2 for glycine. Yeah, there's multiple codons for glycine. There's going to be multiple codons for almost all the amino acids. All right, so UAA is a stop. Let's hit stop. Good. Okay. So this is an exercise, and we can either do this together or you can do it on your own. I think you guys kind of get it, though. Use the table, the genetic code, to deduce the codons that correspond to each amino acid. So if you have the codon chart in front of you, you would be able to see that serine is coded by a series of different codons, right? So if I want serine in my protein, I have a choice of codons that I can use. Same thing with these other amino acids. The major point here is to understand that each codon codes for a specific amino acid. However, you can get the same amino acid using different codons. This becomes important when we talk about mutations. Mutations are a change in the genetic code. Some of those changes will change the amino acid that you get in your protein. Some will not. And when we get into topic 3, we'll talk about that in a little more detail. So, here are some amino acid charts. I'm going to leave this in here for you. You can practice this on your own. And then you can check the answers on your own. You can go back in the recording and look. How do you feel about this? Do you think you'd be able to do it? Should we do it together, or should we go? Give me a thumbs up if you want to go. Give me a thumbs down if you want to stay and do this together. Grant's like, "Yup, we need to do this." Oh, you don't know how to do a thumbs down. There's a thing where you can do a thumbs down. There's no thumbs down in your emojis, really? Let me see. That seems stupid. Oh, I guess I can't do it because I'm the host. And I'm the speaker. Wait, maybe I can. Okay, well, how about this? Thumbs down is too triggering for your generation. Ha! Ha ha. That's all right. I just found out that, like, skinny jeans and a side hair part are, like, Trey millennial, and that's old now. And now I don't know what to do with myself. I hate skinny jeans, though, so it's okay. All right, so how about this? Tell me, would you like to go over this, or would you like to skip it? How about that? You can type it in the chat. Amy says skip. How about I do 1? I'll show you the answer, and then you guys can do the rest. Oh, great says let's go over it for the sake of recording. Okay, all right, good enough. So what you would do is, here's your RNA strand, right? And the first thing I'm going to do, because strings of letters and numbers sometimes mess me up, is I know that I need to be reading codons, so I need to be reading in 3s, right? So the first thing I'm going to do is I'm going to go like this. And then I'm going to use the amino acid chart, and I'm going to figure out what amino acids go with those codons. So let's see. So somebody else that has the amino acid chart in front of them, because I don't, tell me. Well, I know the first one. The first one is methionine. And it's the same for all of these, right? Okay. Actually, the answer is going to pop up underneath, so why don't I put my answer up here? Okay, so somebody tell me the amino acid for CUU. Amy says leucine. LEU. Somebody check her and see if that's right. LEU. Yep, Hannah verifies. Okay, so now we're looking for GGG. Amy says glycine. Somebody check her. I believe her, but we should check her. James says that's correct. Okay, next we need AAA. James says lysine. Okay. Amy says yep. Now we need CAA. James says glutamine. Amy says yep. And the last one, UGA. James says stop. Stop. I don't want to do this anymore. Look at that. All these say stop. And all these say start. Okay, let's see how we did. Pretty good, except for I got the glutamine symbol wrong. But other than that, we did perfect. Okay, now how do you feel about this? Do we need to do the next two, or should we move on? Okay. I'm seeing lots of move on. All right, good. So you guys can do these two for your homework, and then you can come back and check yourselves. Okay. So let's watch all this happen in real time, because this process happens astoundingly fast. It's kind of freaky how fast it happens. Let's watch.
When the RNA copy is complete, it snakes out into the outer part of the cell.
Okay, so this is the nucleus, right? These are those nuclear pores. This is an mRNA molecule that's leaving the nucleus. Now it's in the cytoplasm.
Then, in a dazzling display of choreography, all the components of a molecular machine lock together around the RNA to form a miniature factory called a ribosome. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. The amino acids are the small red tips attached to the transfer molecules. There are different transfer molecules for each of the 20 amino acids. Each transfer molecule carries a 3-letter code that is matched with the RNA in the machine. Now we come to the heart of the process. Inside the ribosome, the RNA is pulled through like a tape. The code for each amino acid is read off, 3 letters at a time, and matched to 3 corresponding letters on the transfer molecules. When the right transfer molecule plugs in, the amino acid it carries is added to the growing protein chain. Again, you are watching this in real time. And after a few seconds, the assembled protein starts to emerge from the ribosome. Ribosomes can make any kind of protein. It just depends what genetic message you feed in on the RNA. In this case, the end product is hemoglobin. The cells in our bone marrow churn out 100 trillion molecules of it per second.
That's crazy!
And as a result, our muscles, brain, and all the vital organs in our body receive the oxygen they need.
Okay. So this information is useful. Now that we understand how this process works, we can leverage that knowledge to help us produce molecules for medical treatment, right? So things like drugs. Before we understood the transcription and the translation process, if you were diabetic, it could very well be a life sentence unless you had insurance, because insulin was extremely expensive. It was expensive because we didn't have a means to produce human insulin economically. Instead, what we had to do is use an organism that had insulin molecules that were similar enough that they worked in humans. And the closest relative that we could do that with that was easy to raise were pigs. So we literally had to raise pigs to harvest insulin to give to people with diabetes. Now, as you can imagine, that process is relatively time-consuming and expensive. However, once we learned how to manipulate this process, specifically how to remove genes from certain organisms and insert them in other organisms, we started using that to our benefit. We took the gene for human insulin and inserted it into a bacteria. That bacteria then divided to form a colony. That colony of bacteria, then with that gene for human insulin, can produce human insulin on a very cheap and very quick pace. Cheap scale, quick pace, right? And it's because of that universality that we talked about. This is really the key to why that works. So here we have a bacteria, and here is a plasmid. A plasmid is basically a circle of DNA. We use restriction enzymes to snip a piece out of that plasmid. We insert the human gene for insulin and coax the bacteria to take that plasmid back by giving it a small electric shock. Then that bacteria divides a whole bunch of times, so now you have an entire colony of bacteria that's capable of producing insulin very quickly. So you basically have 1 million tiny factories for insulin. Then we can refine that insulin and purify it and use it for distribution for diabetes patients. Now, it's the universality of the genetic code that allows this to happen. And this is why diabetes is no longer associated with such high death rates. So if you have a person in your family that has diabetes, they're still around thanks to this process. So here's another person basically saying the exact same thing that I just said, but with a British accent, so it's a little bit cooler.
In this video, we're going to talk about genetic engineering and how it works. This video is a first for me because it's to do with GCSE biology, and this is Unit 1. So, our first step, we need to find a gene that we want. So we're going to find the desired gene somewhere in the DNA of an organism. And in this case, we're going to look for a gene that produces insulin. So insulin is a really important hormone to do with regulating blood sugar levels. And once we've found that gene, we're going to cut it out. And we're going to cut it out using these special enzymes called restriction enzymes. And they sort of work like a chemical pair of scissors. They snip out the DNA in a very particular place. So there we go, we've removed the gene for insulin production, and now we can throw away the rest of our DNA and concentrate on something else. So what we're going to do, we're going to take what's called a bacterial plasmid, and this is a circular ring of DNA that we find in some bacteria, and we're going to cut that plasmid with the same enzyme. The reason why we do this is so that we remove a very particular section of that plasmid that our gene that we want to replicate is going to fit beautifully into. And that's stage 4. So we're going to insert the desired gene into our cut bacterial plasmid. In it goes. And then we're going to insert that whole new modified plasmid into a bacterium. And we do this by giving it a little bit of an electric shock. And that causes the bacterial plasmid to be taken up into the bacteria itself. Once we've done this, we can just let the bacteria divide. And once it starts to divide, it's going to start producing insulin. Now, the more times it divides, the more insulin it's going to produce. So this is a really effective way of producing huge amounts of insulin in a very short amount of time. And that's it. That is genetic engineering of insulin.
So that's kind of it. I mean, we're going to talk about that more in detail when we get into topic 3, and we'll hopefully be doing labs that actually, initially we were supposed to actually do real transformation labs in the classroom, but I think due to current health considerations, we will not be playing with live bacteria in the classroom. So instead, we'll probably do a virtual lab. So at this point, you should be able to do these things. You should be able to define translation, outline how the ribosome reads mRNA and synthesizes polypeptide chains. You should be able to outline the structure of tRNA molecules. You should be able to outline the steps needed to interpret mRNA with an amino acid chart. And you should be able to explain the universality of the genetic code using insulin as an example. So, your notes for this particular segment. You don't have to turn in your notes for translation. What I would prefer you do is turn in this one-pager that reviews transcription and translation. Make sure that you include these things. My hint is to work backwards from a protein. So go find a real simple, tiny little protein that doesn't have a lot of amino acids, or make one up. It's fine with me. And work backwards. But before you do this, I have some models for you to look at that will help you visualize this process step by step. So I'm going to discard that, and we're going to go like that, and we're going to go like that. So let's see. If we go to transcription and translation models. So if you click on that, click. Okay, so you have a model of transcription here. This will take you through the process of transcription. Prepare for transcription. And then you're going to, let's see, where is our complementary RNA strand? So let's see, G, C. I guess they don't start there. T, that will be A. There we go. So T, A, it starts there. So then you click and you go through the process of creating a strand of mRNA. And that's transcription from DNA to RNA. Next, you have a translation model where that same strand of mRNA that you just created, you're going to use that and translate it into a protein. You're going to do that. You can click translate and do the whole thing, or you can do it yourself step by step. And this will help you visualize that process. Once you're done playing with these models, and you can take as much time as you want to play with these models, there is a quiz. Let me go to the student view. Hold on just a second. If you hit next, there's a quiz. Now, don't freak out because the models are embedded in the quiz. So you can use the models while you're taking the quiz. So here are the questions, right? And then the models are in here. It says use the model to answer questions 1 through 5. And then use the model to answer questions 6, 7, and 8. Okay? And then there's a couple questions down at the bottom. This is another type of codon chart. You just read it from the center out. Okay? All right. So you are free to go. Enjoy yourself. If you have any questions, let me know. And I'll see you guys next time.